Good evening, everybody. My topic is Holiday model. This is a symbol model which is useful for understanding the steps of homologous recombination. It is clear that most recombination events involve synthesis of new DNA, a feature absent from this model, but still it is useful for understanding the recombination process. This model is discovered by Robin Holliday in 1964 in E. coli. It explains the heteroduplex formation and gene conversion during recombination. And also it illustrates the critical steps of pairing homologous duplexes, formation of heteroduplex, formation of recombination joins, branch migration, and resolution. The process starts with breakage at corresponding points of homologous pairs of two paired DNA duplexes, and this breakage allows movements of free ends created by the next. Each strand at broken end leaves its partner, crosses over displacing broken strand, and pair with its complement. It creates connection between two DNA duplexes, and this connection is sustained by hydrogen bonding. The point at which individual strands of DNA crosses from one duplex to the other is called as recombinant joint. At site of recombination, each duplex has a region consist of one strand from each of two parent DNA molecule, and it is known as heteroduplex DNA. The joint molecule formed due to exchange of broken strands need to be resolved from each other into two separate DNA molecules, and the process is called as resolution, and it took place in two steps. In first one of the two DNA duplexes in the joint molecule undergoes rotation and give rise to a planar molecule. And in second step, in planar molecule, there are four strands and two of them are nicked, and the consequences of nicking will depend upon which pair is nicked. And this is a diagram that shows the Holliday model, and it includes the several steps. In A, we can see two homologous double-stranded DNA molecules aligned with each other which have different alleles of same genes. In A, we can see the homologous double-stranded DNA molecules. And on both DNA duplex, single-stranded nicks are formed. That is, in B, we can see that nicks are formed. Single-stranded nicks are formed on the same position. In the same position, the single-stranded nicks are formed. Then strand inversion occurs. So after the nicks are formed, the strand inversion occurs. In this step of homologous recombination, initial short region of base pairing form between two recombining DNA duplexes near the nicks, and Holliday junction is formed. So in C, we can see the Holliday junction is formed, and it is the region where two DNA duplex form a cross-like structure of two DNA strands. And here is by two DNA strands a cross-like structure is formed, and it is called as the Holliday junction. After the Holliday junction is formed, the branch migration occurs. That is, the Holliday junction is moving. It is moved by melting and forming base pair repeatedly. In parental DNA molecule, base pairs are broken. So the branch migration occurs after the Holliday junction is moved, and the branch migration is the where in which the Holliday junction moves by melting and forming base pairs repeatedly. In parental DNA molecule, base pairs are broken, and in recombination intermediate, identical base pairs are formed. The DNA duplexes have very small sequence differences as they have different allele of same genes. So after branch migration, the new DNA duplex will have a few sequence mismatches at those regions. So after the branch migration has occurs, the new DNA duplex will have a sequence with some mismatches, and that region is called as heteroduplex region, and thus the DNA is called as heteroduplex DNA. In E, we can see the heteroduplex region. Okay. And at the last step of the DNA recombination, the Holliday junction is cleared to separate DNA duplexes, and the process is called as resolution, as I told you earlier. So for better understanding, the Holliday structure is being rotated into a planar structure without crossing strands. We can see that the Holliday structure is being converted into a planar like structure for better understanding. So, and the resolution process has two possible sites. It has two possible sites. Each site can give different outcome after cleaving. So, after cleaving, each site can give different outcome. So, in the first cut site, that is vertically, vertically cutting. As a result, original DNA duplexes are spliced together in resulting duplexes, so they are named as resulting duplex will have one pair of identical allele from one parent. That is in F, we can see that in F, that is the it has the identical alleles from one parent and a region having heteroduplex DNA. That is in Fe, it thus is the heteroduplex DNA and a pair of allele from other parent in last region. D allele switched from one duplex to another, so called crossover region. D, it has been switched from one duplex to another, so it is called as crossover region, and thus the process and the result just attained is called as the crossover product. And the second cut is like the horizontal cut, that is like here, like this. In the horizontal cut, as a result of that, the product have a patch of hybrid DNA. So they are called as patch recombination product and also known as non-crossover product. As the D and F has not changed in their position, we can see in G, the D, yeah, the D and F has not changed in their position in new product and remains identical like a parental duplex. So this is the explanation for the diagram of the Holliday model. And the gene conversion, the formation of heteroduplex segments can provide the explanation for gene conversion. When the heteroduplex is formed, it can provide the explanation for the gene conversion. When the heteroduplex is formed, it is when it is homozygous, it will not be subject to any gene conversion, that is no mismatch. And if the region that is heterozygous, then there will be mismatch between the two strands of DNA molecule. Heteroduplex formation region, when it is if the heteroduplex formation formed region is homozygous, there will be no any mismatches. If the region that is formed is heterozygous, then there will be mismatch between two strands of DNA molecule, and it may lead to gene conversion in either two directions. So the phenomenon of gene conversion provide clues for explaining the genetic recombination at the molecular level. As the Holliday model has been used, it has some shortcomings, like the specificity of the enzymes in eukaryotes is not fully understood, and also the recombination is not known to begin at double strand break or single strand gap in one DNA molecule. So I hope you all understand my video, and thank you.